Hello everyone, welcome to the Dandelion Biology series. Today's video will be about topic 1.4, membrane transport under the cell biology unit. This will be an extension of what we talked about last lesson, where you learned about the structure of the cell membrane. So as a recap, the cell membrane is an organelle that regulates what goes in and out of a cell. The current accepted model for the structure of the cell membrane is the fluid mosaic model proposed by Singer and Nicholson. Due to the structure of the cell membrane, it has two key properties. One, it is semi-permeable, meaning that only certain things can pass through. And two, it is selective, meaning that it can regulate the passage of materials. Membrane transport can be either passive or active. Passive transport does not require any additional input of energy, while active transport requires energy in the form of ATP. First, let's talk about passive transport. Passive transport does not require any energy as it relies on the naturally occurring process of diffusion. Diffusion refers to the net movement of particles from an area of higher concentration to an area of lower concentration. One thing to note is that this is the net movement, meaning that random particle movement will still happen both ways, but the general movement follows the direction of the concentration gradient, which is towards the region of lower concentration. Particles will move along the concentration gradient until equilibrium is reached, or when there is equal particle movement to both sides. There are two types of passive transport designated for different types of materials. First is simple diffusion, which happens directly through the plasma membrane, and this is only possible for small or lipophilic molecules. Some examples include oxygen, carbon dioxide, water, and steroids. The other type is facilitated diffusion, which is used for substances that can't freely pass through the cell membrane, such as charged ions and large macromolecules, especially polar macromolecules. Some examples include glucose, sodium ions, and potassium ions. These substances can diffuse through the cell membrane via channels or carriers made out of proteins. Protein channels have hydrophilic pores which allow ions to pass through, and carrier proteins undergo conformational changes as it binds to specific substances and it translocates them across the membrane. Osmosis is a specific type of diffusion that involves the movement of water molecules. In cells, when there is an imbalance of solutes between the two sides of the cell membrane, water will diffuse through the membrane to balance the solute concentration, as the solutes themselves cannot pass through the membrane. Osmolarity measures solute concentration, which can be classified with three types of tonicities. Hypertonic refers to when a solution has a higher relative solute concentration. When a cell is put in a hypertonic solution, water will diffuse out of the cell and into the solution, causing it to shrivel. Hypotonic is when a solution has a lower relative solute concentration. In a hypotonic solution, water will diffuse into the cell, causing lysis. Isotonic is when a solution has the same relative solute concentration. If a cell is put into an isotonic solution, the net movement of water will be zero. Here is a visualization of what would happen if animal cells and plant cells were put into solutions of different tonicities. Unlike animal cells, plant cells will maintain an overall fixed shape due to their rigid cell wall. Moving on to active transport. Active transport is used when cells need to intake materials against a concentration gradient. This requires energy input in the form of ATP. Carrier proteins, also referred to as protein pumps, are used to mediate this process. We know that carrier proteins engage in passive transport when they move substances along the concentration gradient, but here, when they are moving against the concentration gradient, they will be a part of active transport. Direct active transport is when a molecule binds to the protein pump, ATP hydrolysis causes a conformational change, and the molecule is translocated across the membrane. Indirect or secondary active transport tags along to this process. A molecule relies on the electrochemical gradient generated by active transport as a source of energy to simultaneously move it across the membrane. When this happens in the same direction as the direct active transport, it is called symport. When it happens in the opposite direction, it is called antiport. The last type of active transport we'll be going over is vesicular transport, which allows for the bulk transport of materials without having to cross the cell membrane. Relating back to the structure of the cell membrane, the fluidity of the plasma membrane allows it to be easily broken and reformed through the hydrolysis of ATP. The cell membrane can separate and fuse to form vesicles that transport a bulk of materials at once. Endocytosis is when materials enter the cell through being internalized within the vesicle formed from the cell membrane. Examples of this are phagocytosis, which is the ingestion of solid substances, and pinocytosis, which is the ingestion of liquid substances. Lastly, exocytosis is when materials exit the cell through vesicular transport. Vesicles are usually created in the rough ER, repackaged in the Golgi body, then sent to exit the cell membrane. And that is all from Dandelion for today. If you have any questions, please comment down below. And if you would like to stay tuned for further videos, please click subscribe. See you next time.